Okay, so it's a real pleasure to welcome Dr. Christine Vande Velde next. So she comes to us all the way from Canada, where she's a professor at the University of Montreal. Christine has had a long-standing interest in ALS research. She actually trained in Don Cleveland's lab, where she focused on the mechanisms associated with misfolded SOD1. In her own independent research program, she's been largely focused on TDP-43 biology. And today she's going to talk to us about a subject that's close to her heart, which is stress granular biology. And I'd like to commend her on sticking with this and trying to make sense of this controversial pathway in the context of ALS. And I'd like to say one more thing, that beyond her robust research program, Christine is committed to the field through mentorship, through providing community service, really. She's the scientific director of the ALS Packard Association, and she plays a critical role in identifying talents, fostering collaborations, and helping facilitating the research for all of us. So thank you, Christine, and welcome.
Thank you, Evangelist. So thank you very much for this really warm invitation, Hande and Evangelos, and the entire Les Turner Foundation team. I really do appreciate the opportunity to speak about something that, yes, I do care quite a bit about, and even though I'm still mourning the fact that the Blue Jays lost the World Series a couple of days ago, I am still willing to be here in the US and talk to you all. Okay, so stress granules and ALS, I titled it provocatively Friend or Foe, and this is pays homage to a recent review that used this title from Gobai in China. This idea of stress granules and how they link to ALS has been hotly debated now for more than a decade, and I'm here today to sort of, let's review some of the data that has been presented to the field and sort of where our contributions might lie as we look at this in an in vivo context. So what are stress granules, for those of you that are not sort of indoctrinated yet into this idea of what these structures are? These are subcellular RNA granules. They are membraneless, and they contain numerous RNA binding proteins and mostly polyadenylated mRNAs that have fallen off at the ribosomes, although there's also several long non-coding RNAs also included in them. It's thought that they're basically supposed to be storage sites of suppressed RNAs, although there has been evidence now that there is translation occurring within these stress granules. And it was also considered that maybe these stress granules, in association with another RNA granule subset called processing bodies, that they might be ways in which RNAs are sorted for degradation into the P granules. However, the P bodies, but however, that has not yet been demonstrated either. Traditionally, these are considered, they're evolutionarily conserved. They were first identified in plants, and in fact, they're thought to be really anti-apoptotic structures, because they sequester various proteins that are related to triggering that particular cell death program. And they've also been implicated in viral implication as an inhibitory mechanism. So fundamentally, what I want you to take away is that stress granules are actually a cell biological entity that actually integrates extrinsic and intrinsic signaling pathways. And so these granules are actually triggered whenever cells encounter some adverse condition. And this can be a viral infection, it could be heavy metals, it could be a traumatic injury. It's really not defined. There are lots of different ways to generate these particular subcellular structures. So how did they become linked to ALS? And that's through a couple of different things. So first, I want to highlight that we talk about ALS. We all know what ALS is and FTD, but we talk a lot here in the room about the genetic forms of these diseases, as we just heard, because that's what we can model and that's what we can study. But we know that the majority of patients, actually, we don't have a firm genetic diagnosis for them, and we assume that there are various environmental factors that are linked, although we don't totally agree on what those are. So from the genetic standpoint, there's more than 40 different genes linked to ALS. Several of these are mRNA binding proteins that are actually localized to these stress granule structures that I described. And several of these point mutations actually enhance their aggregation, or what's also referred to as liquid-liquid phase separation, this idea that these mutated proteins can sort of self-associate into larger order structures. In the context of the environment, as I mentioned, we don't really agree on what the different hits might be, but we've definitely, there's been many models put forward with the idea that you have a combination of genetic load, our aging, and various environmental exposures that can contribute to an onset of ALS. So how did TDP-43 and stress granules and how did that make that link to ALS? It comes from images like this. And so this is some of the original pathology looking at TDP-43, which we all know is supposed to be normally in the nucleus here with a little bit of it in the cytoplasm, probably shuttling actively, participating in distribution of RNAs throughout the cell. But in the context of ALS and FTD, and now we know Alzheimer's, Huntington's as well, is that TDP-43 sort of exits the nucleus and seems to localize to various other cytoplasmic structures. And so sometimes they're these large round ones, sometimes it's much smaller granular ones, sometimes there's these big skein-like things. This happens in about 97% of all cases and about 50% of FTD cases. And just as a small reminder, there is evidence of TDP-43 genetically being linked to ALS through a variety of different mutations that seem to cluster within the C-terminus. But this really accounts for a real minority of all ALS patients. It's this immunohistopathological signature of TDP-43 that we talk about most often. So in the context of stress granules and TDP-43 pathology, it's been put forward that perhaps some of these structures actually might arise from RNA granules that form naturally within cells, and so particularly from the stress granules. And so just to kind of, in a cartoon fashion here, just sort of show what the normal life cycle of an RNA, of a stress granule is that normally you have RNA binding proteins and associated polyadenylated RNAs that were kicked off of the ribosome in response to a stress, and that happens through a phosphorylation event at EIF2 alpha, so that polyadenylated RNAs get stored, allegedly, and that gives ribosomal priority to other things that the cell might need to get through whatever this stress event is. This collection of RNA binding proteins and RNAs are going to self-associate and undergo, like we'll look at phase separation, and they sort of undergo like little droplets into bigger droplets. And so if you think about the vinaigrette that you make for your dinner, you have little tiny droplets in emulsion that then get bigger and bigger. And it's thought that these large ones are where the activity of the stress granule lies. Once the stress is cleared, these things basically fall apart, just like in your vinaigrette, they fall apart. And there are different mechanisms that have been proposed for that, but basically these structures resolubilize, and those RNAs that were originally held there now return to the ribosome, and the cell basically resumes business as normal. In the context of ALS, what's presumed is that perhaps that this pathway, this life cycle is not working as well, especially in the disassembly phase, such that these granules will transform into the aggregates and that these actually are toxic. And there is lots of evidence to show that aggregation of TDP-43 is toxic. Alternatively, we would propose that perhaps this mechanism just doesn't happen at all, is that there's a failure in the system in order to go that initial phase separation event, and that this would also then imply that there's neuronal vulnerability. So if we review the evidence about the gain of function, so there are many different reports of aggregations of stress granules within postmortem material. This one here was one of the first, showing TIA1, which is considered to be a canonical stress granule marker, as co-associating with TDP-43. This was actually only shown in this one paper and has never been replicated. There's also dozens upon dozens of papers that have shown TDP-43 to be co-localizing with stress granules following different types of stressors and different types of cell systems, including neurons, HeLa cells, U2OS cells, but almost all of those are actually been exploiting overexpression systems. Very few have actually looked at it in the context of physiological TDP-43 expression. And using those type of models, actually there have been many different drug screens that have been used to try to look at whether or not we can devise ways to inhibit stress granule formation. And so that those have been published by many groups, and those are being used to fuel sort of drug discovery programs around all over the place. And this idea that if you prevent stress granule, TDP-43 recruitment to the stress granules, that you can ultimately prevent cell death. However, if we consider the loss of function pathway, first I want to draw attention to this work. This is actually from Don's group, in which they show that TDP-43 assemblies are independent of canonical stress granules. You can see that we basically get red and green dots. There's originally initially an overlap between TDP-43, which is expressed here. This isn't a CRISPR-tagged version of TDP-43, so endogenous expression levels with a CRISPR-tagged stress granule marker. And you'll see that there is a co-localization of the green and red signal that then separates, which actually shows that the TDP-43 and the stress granule is actually physically separate entities. And another paper that was published around the same time, using patient material, actually it was shown that there was no polyadenylated RNA within the TDP-43 inclusions shown in patients. And this is important because polyadenylated RNA is actually an obligated component of stress granules. And some work from my group that we've shown in sort of various cell models at this point, this is from HeLa cells, early days, in which we actually showed that if you knock down TDP43, you actually get a loss of the G3BP1 protein, and this is going to become important because this is actually a core stress granule assembly factor, and its paralog G3BP2 is completely immune to TDP43 manipulation. And this is showing the consequence on the stress granule pathway here. This is in response to arsenite, which is highly artificial, but we see that normally you would get those little droplets that I mentioned that get coalesce into larger ones, and we don't see that so well in the context of the TDP knockdown. We've also shown that G3P1 mRNA is actually selectively downregulated in those neurons that actually show TDP43 pathology. So here, if you focus on these two neurons, which show cytoplasmic TDP but no nuclear, we have a loss of the red signal, which are reflecting on individual G3BP1 transcripts. And then in other work that was published independently using single-cell mass proteomics is that we see that G3BP1 is one of the hits that is actually downregulated in neurons that are lacking nuclear TDP43. And again, to highlight why G3BP1 is going to become a focus of this talk in particular, is that it actually sits really at the nexus of stress granule assembly. It is considered the core, and it's the G3BP1 and its partner G3BP2 are the only two proteins that are absolutely required for stress granule assembly. Very, very recently, there was work from Peter Todd's group that suggested that maybe knocking down stress granules or inhibiting stress granules might actually worsen phenotypes, and he showed that both in TDP and C9 Drosophila models, and as well as in some IPS neurons. And then I want to highlight here that unfortunately, just this year, we've had two major phase 2-3 trials that have failed that were actually aiming to prevent stress granule formation as a potential therapeutic strategy. And this work was informed by those drug screening strategies done that I mentioned earlier. So the summary of the evidence that sort of links stress granules to TDP43 pathology, I classify it comes down into two categories. Stress granule dependent, meaning there's a gain of function that leading to TDP43 pathology, and then the stress granule independent. And I want to highlight that one of the big differences between these approaches is that in the gain of function, they really relied on overexpression systems of TDP43, and the presumption that all the visualized structures actually are stress granules without actually validating that they do contain polydendulated RNA, whereas in the stress granule independent sort of side of things is that it's primarily based on physiological expression of TDP43. It has support from both patient data and from tissues from patients. And so how do we actually get at this relationship between stress granules and TDP43 pathology in vivo is something that I was really become very passionate about because I just didn't love the overexpression studies that had been done, and how do we get that, to make that link between how does it really impact disease. And so we set about in the lab trying to figure out a way, could we model some sort of stress response in mice. And so this is a cartoon that I borrowed from the internet that talks about various stress factors or environmental factors that have been related to ALS. There's no good consensus about any of these, so please give me a grain of salt here on this one, because I think there's just as many papers that support sort of, there's an increased risk of ALS as a result of excessive exercise, as we see in so many professional athletes, but there's also many papers that say that that's not true. However, if we kind of break it down and we look at these various possibilities, one of the things that sort of came to light after lots of discussion in the lab and with various individuals, including I do some work for the Canadian Defence, which I know that sounds kind of like an oxymoron, but there is a military in Canada. That's about to get improved, apparently. There is a lot of discussion about exertional heat stress and sort of acute heat stress that soldiers might experience during their training or in theater, and this idea that this is a physiological disruption to their system that equates to stress. And so I do think that some of the evidence, both in Canada and the US, that there is a link between military service and ALS is true. And so we used heat as a particular way to maybe how could we invoke stress in a mouse model, and because obviously we can't put them through boot camp. So I'm going to tell you a little bit about a mouse model that we designed in which we are using a TDP43 mouse as well as regular black 6 mice that we exposed them to an acute hyperthermia. So it's 20 minutes at 44 degrees. It's not at all related to ALS, as I said. It's just as a way to try to mimic some sort of environmental stressor. And so if we expose these mice to 20 minutes of high heat, we actually can see that we get phosphorylation of VIF2 alpha, which demonstrates that we've actually engaged the integrated stress response. If we now look for stress granules, and now we're focusing here on the spinal cord motor neurons of these mice, we can see here that in the control, there's no partitioning of the HUR signal in the cytoplasm, whereas we see very clear, nice, round puncta that label positively for HUR, suggesting that there are stress granules. We've used many other markers, and you'll see that all of these also shows a partitioning of the cytoplasmic signal. Although none of them show there's really nice droplets and they don't show a uniform pattern like we're sort of conditioned to expect in the in vitro cell-based experiments. This caused us quite a bit of concern, and we said that we switched to looking at the RNA, and so we see very clearly that if you have you look at the cytosolic polyadenylated RNA, you see very clearly in the stressed animals, you get a partitioning of the signal, and so going forward, I'm going to show you images that are really focused on the RNA localization rather than the protein, since there's such a disparity in the protein signal. So what happens if we apply this model now to ALS mouse models? And so we've taken two different mouse models. The first model I'll introduce to you is the M337V mouse from Kevin Talbot's group. It's a knock-in to the ROSA 26. You can see that there is a very slight overexpression of the human TDP43 and that the mouse allele is maintained. And in fact, as these animals age, the autoregulation is maintained so that total levels of TDP43 are not disturbed. These mice have a very subtle phenotype, very, very minor NMJ defects like at 12, 18 months. Importantly, there is no evidence of TDP43 nuclear inclusions or clearance. And then we also use the G93A mouse, the SOD1 model, because also there is no TDP43 inclusions or nuclear clearance in that mouse either. So these are ideal models in which to evaluate the interplay between stress granules and TDP43 inclusion formation. And so just because this mouse, the M337V, is probably not familiar to many people in the group, we looked to see whether or not it was a gain or a loss of function with respect to splicing. And so this is just our positive controls here, in which you can see the inclusion of a cryptic exon or a skiptic exon. And we see that this mouse is neither a gain or a loss, suggesting that the TDP43 levels are really not changed. We have preserved physiological levels of TDP43 in this mice with respect to its function and splicing. So the first thing I want to demonstrate to you is that in these mice, expressing almost physiological levels of TDP43 mutation is that we do not see any evidence for stress granule formation. Again, for focusing here on the cytoplasmic polyadenylated RNA signal, we see completely homogeneous, whereas if we look in the G93A mouse model, we have a very nice formation of stress granules. You can see visualize very beautifully in these neurons. And so if we quantify that up, so that's just looking at the cells, or we use a MATLAB script to look at granularity, we see very clearly that the M337V mouse model is actually deficient in stress granule formation compared to its littermates or the G93A. And this is despite the fact that we have equivalent engagement of the ISR response as judged by the IF2 alpha phosphorylation. So whatever we're seeing is downstream. So what happens with TDP43? So this is just spinal cords, and we're looking at the motor neurons. These very, very large nuclei are actually motor neurons, and we're looking to see what happens with TDP43. And so we see even in the black 6 mice that we have some mislocalization of TDP43 that can happen in response to this hyperthermia, suggesting maybe this is a physiological response, and it seems to be exacerbated in the presence of a TDP43 mutation, but is relatively minor in the context of the G93A. So we're seeing stress-induced TDP43 nuclear clearance here that's exacerbated by the mutant allele, but we do not see inclusions. What about other TDP mouse models? And so I'm going to include data here for you with two other models. So this is a mouse model made by Jamin Shriharan using the Q331K, as well as a mouse model that's a doxycycline-inducible expression of TDP43 that's sequestered in the cytoplasm, the delta NLS model. And so these mice do not generate cryptics, so they are not loss of function, but they do show inclusion of a skiptic exon, supporting the evidence that's published that these are both gain of function models. In both of these mouse models, which have one which features robust cytoplasmic TDP43 aggregates and one that doesn't, we see that there's actually no effect on the stress granule formation of these two different models. So there's something really different about the M337V mutation. And interestingly, here in this one, the Q331K, we see a similar level of TDP43 nuclear clearance after the acute hyperthermia, regardless of the phenotype of the genotype. So I'm just going to summarize here in cartoon form a bunch of other work that we looked at trying to explain where was this deficit of stress granules, and what we noticed was there's actually a decrease in the expression of various RNA binding proteins in the M337V mice, including G3BP1. We saw that cytoplasmic mRNA levels in general also were down, and we saw an enhancement of the chaperone EIF4A1, which is demonstrated to actually block stress granule formation. And so we sort of make a schematic about the idea that these three things combined actually prevent TDP43, or prevent the stress granules from crossing this percolation threshold. And so we're lacking the formation of stress granules in these mice. What's important to note here is that we had no stress granules and we had no TDP43 inclusions in these mice. And we appreciate that there are a lot of limitations with such an acute model. And so, for example, it doesn't allow us to really evaluate whether or not the persistence of stress granules is actually what are seeding TDP43 inclusions that are observed in patients' materials. So we went back to the drawing board a little bit and thought a little bit more about how could we model that. And we thought, well, in an individual, we probably have recurrent exposure. And so we would go through multiple cycles of stress granule assembly and disassembly. And so is there a way that we could model that? As well, the one thing that we definitely universally agree on is that aging is the #1 risk factor in ALS. And so what would happen if we combined recurrent stress exposures using heat and aging, and how would this impact stress granule biology, and importantly, how does this relate to TDP43 inclusions? And so the first data that I would present to you is that we actually observed that there was an impact of aging on stress granule biology, such that while this response is very robust in the younger animals at 4 months, it is really lost over time, that in a normal black 6 mouse at 18 months is that we're actually lost. The cells have lost the capacity to make stress granules by about 50%. And so this is important when we think about ALS as an aging disease. Can we model stress granule dynamics in mice? And so the previous model I showed you was using acute mice, in which mice were subjected to stress for the 20 minutes. Here we gave them a longer stress, and then we gave them a break, and then we stressed them again. And what we can see is that we can see the stress granules assembly and disassembly occurring in these mouse models, which gives us an opportunity now to look at recurrent stress cycles in the mice. So this is just the quantification of that. So we took mice that then we looked to see whether or not we saw stress granule formation after applying multiple stress cycles, and we see again that in the TDP43 mice we're not seeing the stress granule assembly compared to the non-transgenic litter mates. What if we took those mice now and we cycled them, we're going to cycle them for a month, twice a week for 4 weeks, and do we see stress granule formation in these mice? And so no, we do not. However, one of the things that we did find is that we saw TR puncta formation that was accumulating over time in these mice, but then it seemed to be something that was lost, in the aged mice, but something that we saw very robustly in the early mice. And so that was curious because it kind of mimicked what was being observed in the non-transgenic mice, showing that there was no relationship between the TDP43 mutation and the presence of these TR puncta. And so this also draws into question this idea that TR puncta, as they're observed, are for sure stress granules, because here we do not see any evidence of polyadenylated RNA containing stress granules, but we are seeing cytoplasmic TR granules. What about the TDP43 nuclear clearance? And so as before, we saw that some proportion of the neurons would show a clearance of nuclear TDP43, but again here we're not seeing in this particular image, we're not seeing a lot of cytoplasmic TDP43. What we saw is that this was something that was happening in the non-transgenics, but was now lost in the context of the mutant, regardless of the age. What about the cytoplasmic puncta? So this idea of the stress granules. So these mouse model doesn't make stress granules. Do we see TDP43 cytoplasmic puncta? And the answer is we do see some in the non-transgenics, and we also see them in the mutant. And so the presence of these TDP43 cytoplasmic puncta are exacerbated both by genotype and age. But this is happening in the context of a mouse model that does not form stress granules, suggesting that these truly are independent events. And here, just to show you that there's no co-localization of TDP43 cytoplasmic puncta with known stress granule proteins such as TR or HUR in this case. And so in this particular neuron, you see that they're really happening at opposite ends of the neuron, whereas in this case, you're seeing that the signals are really separate in the same region of the neuron. So there's no co-localization with these markers. What about, how does this relate to motor neuron loss? And so if we look in mice that we've stressed for a month, and we either look at them right away or we look at them 4 months later after recovery, do we see any motor neuron loss? And so you'll see here in this particular, as I mentioned before, this particular mouse model doesn't really show any motor neuron loss. It looks similar to the non-transgenic litter mates. However, when we allow the mice to recover for 4 months post-stress, we start to see that there's some evidence of motor neuron loss. And actually, if you zoom in on this, you start to see that what we're actually seeing is alpha motor neuron loss, which are the neurons that are selectively lost in the disease, and we do not see any evidence of gamma motor neuron effect by this stress paradigm. So in this mouse model that originally, as I mentioned to you, has no motor neuron loss, what we have here is actually a selective and progressive induction of motor neuron loss that can be brought on by aging and exposure to multiple stress cycles. So I think that the summary of the evidence that I've shown is that the TDP-43 inclusions are stress granule independent, and we've integrated the risk factors of genetic predisposition and environmental stress and aging here. We're showing that there is an impairment of stress granule formation in motor neurons of mutant TDP-43 mice that have near physiological levels of expression, and this is a really important point that I can't underline enough, is that all of these granules are very sensitive to stoichiometry. Stress-induced TDP-43 cytoplasmic puncta are independent of TR, and so you can have the presence of those, but there's no link to the disease at all, even though they can be exacerbated both by age and genotype. And we see a selective loss of alpha motor neurons that correlates with TDP-43, but not TR. So we're seeing motor neuron loss despite the fact that there are TR puncta. So I just want to come back to some sort of the digging into the molecular in the last few minutes that I have, is that we've previously published that TDP-43 actually stabilizes the G3BP1 mRNA. That was part of that data that I showed at the beginning, showing that G3BP1 transcripts were lost in the neurons that have a TDP-43 pathology. And so we actually, using a variety of molecular biology approaches, we actually isolated that there's a very highly conserved sequence that's sitting here in the 3' UTR. TDP-43 binds this. And if you knock it out, if you take away that sequence, you actually normalize the stability of that transcript. And we've now got data that actually we've isolated that there's a neuronally enriched microRNA that is actively competing with TDP-43 in this region, and that work is coming soon. The other thing that I want to highlight, though, is focusing here on these other TDP-43 binding peaks that are sitting in an intronic region. And so in work that we and others have done, we've actually concluded that this is actually a cryptic exon. And so if you do TDP-43 knockdown in neurons, you'll actually see this little cryptic exon that pops up consistently, both in our work as well as work from Pietro Fratta and Michael Ward. And if you dig into this cryptic exon has canonical 5' and 3' splice sites, and it's located immediately upstream of the TDP-43 binding site. And actually, this cryptic exon does show up in Pietro's work that was published a couple years ago. So what is this cryptic exon? Should we care about it? I originally didn't want to because I didn't think that it was going to have relevance, but that cryptic exon actually is in frame. It generates a 10 amino acid in frame insertion, and so the first question is it something you can detect in patients? And this is work with Alyssa Coyne, in which she's used a panel of over 100 different IPS spinal motor neurons lines, and those that have a TDP-43 signature, a splicing signature as she's published elsewhere, they also show a high inclusion of the cryptic exon for G3BP1, as do the C9s, which also have TDP-43 pathology. We have only 3 lines with TDP-43 mutations, but we also see that there seems to be an inclusion of the cryptic exon there as well. If we look in the New York genome data, we actually see that the cryptic exon seems to also be included in those samples that motor cortices and frontal cortices from patients that have ALS with TDP-43 pathology, but not so in the controls or those patients that lack TDP-43 pathology. So the cryptic exon, as I mentioned, it's a 10 amino acid peptide that is inserted in frame. It inserts immediately into the NTF2L domain. This domain is exceptionally important because it actually is what governs stress granule dynamics. It actually sits, this is an alpha fold projection showing where this peptide would sit. It actually disrupts the binding with caprin-1, which we can demonstrate here very clearly using a co-IP experiment in cells that are lacking the endogenous G3BP1 and 2. And so just to come back to this picture showing that G3BP1 sits at the node, is that caprin-1 is one of the major interactors here. And so just again, this cryptic exon does not exist in G3BP2, and G3BP2 is absolutely not responsive to TDP-43 nuclear levels. We also did an apex labeling approach showing that when we express the cryptic version of G3BP1 in cells that are lacking the normal complement of G3BP1, we see a loss of various stress granule proteins as marked in red here. We also did some experiments in which we tried to overexpress the G3BP1, the wild type or the cryptic. These are stable cell lines that are expressing fairly low levels, but still nonetheless it's overexpression, and I appreciate the challenge there. But we see that in these cells, which are U2OS knockouts for G3BP1 and 2, the wild type can fully complement and restore stress granule formation in response with arsenite, whereas the cryptic one does not. And we saw a similar sort of thing with heat. And now if we use the U2OS cells, which have the parental cells which have normal G3P1 and 2, if we express the cryptic, what we actually see is in this cell that's expressing the cryptic, we actually block stress granule formation, suggesting that it's functioning as a dominant negative in this context. So this cryptic G3P1, one of the things I didn't tell you about is that it's not expressed in neuronal cells, which causes a lot of grief at the beginning. So using U2OS or HeLa cells, which are common cell lines used to study stress granule biology, you do not detect this cryptic when you knock down TDP43, but we can detect UNC13A, which is curious because that's a neuronally expressed, it's traditionally thought to be neuronally expressed, but we can find this in these cells. So it got us thinking a lot about why were we detecting that cryptic in neuronal cells and not non-neuronal cells. And so my postdoc, Hannah Fackham, who's a really smart RNA biologist, really dug into the literature about what could be differences in splicing factors. And so Hannah uncovered SRM4 as a neuronal RNA binding protein that actually, very interestingly, had a number of binding sites that were sitting just immediately around the cryptic exon as well as in between the TDP43 sequence. And so now if we overexpress SRM4 in U2OS cells, we can actually, with a mini gene, we can drive the insertion of this cryptic exon. And actually, if we look at the endogenous, if we make a stable cell line that's inducible for SRM4, we can see that with increasing expression of SRM4, we now can drive the expression of the endogenous G3P1 cryptic, and we don't touch TDP43. So we've actually found the enhancer that governs G3P1 cryptic splicing in combination with TDP43 repression. So we present data here that there are two different mechanisms by which G3BP1 can be regulated. The RNA stability mechanism, which I demonstrated many years before, and now this recent one using splicing. And in the context of TDP43 pathology, really TDP43 nuclear clearance is that we can see that there's a dose-dependent, I didn't show you the dose-dependent data, but there's definitely a dose-dependent effect that when TDP43 nuclear clearance is a little bit, you can get inclusion of this cryptic exon that's driven by SRM4. And if you knock down TDP43 a lot, or you really clear the nuclear pool, you actually can, you end up with microRNA-mediated decay of this transcript. Both of these, sort of all roads lead to Rome here, all of them lead to an impairment of the stress granule pathway and neuronal vulnerability and loss, which we've shown elsewhere. So just to summarize, we have two different mechanisms governing TDP43, G3BP1 that TDP43 is acting through, and we have an in vivo model that favors stress granule independent for TDP43 pathology. We think that they're really unlinked in the in vivo context. And in this particular mouse model, we actually demonstrate that if you impair stress granule formation, you actually do have a loss of motor neurons. And we have a more direct experiment ongoing now in which we will use a viral peptide that will inhibit stress granules in mice and actually acts directly about motor neuron loss in a different way. And I just want to remind the field sort of that G3P1 knockout mice exist, and they're actually embryonic lethals, and their primary phenotype is that they've lost their neurons. So the cortices are completely wiped out of their of the pyramidal neurons, as well as striatal and Purkinje's. And so I'd like to propose that the replacement of stress granules should be a therapeutic strategy more so than the inhibition of stress granules. So with that, I thank my team. All of the in vivo stress granule work has been led by Alicia Jabinski, who is a very talented graduate student who's about to move to Michigan for her postdoc. Hannah Fackham, a postdoc who's really an authority on the RNA stability and the splicing, and she'll be looking for a faculty position in the coming years. All my collaborators, I'm really grateful, and as well as funding, and thank you for your attention.
All right.
Can I start with the questions on the chat?
Sure.
Mm-hmm. Okay, thank you so much. So we have a couple questions on the chat. They all liked your presentation very much. And Ron Newberry asks, can you please give us some details about the derifenacin trial?
About which trial?
It's a clinical trial. I think it's called derifenacin.
The DARI trial?
DARI trial, yes.
Because I'm from Montreal, I guess. This is a trial from Richard Robitaille, my colleague at the University of Montreal, and it's a muscarinic receptor agonist, and that trial is enrolling, as I know, in Montreal, and it's really looking at the NMJ stability,
Yes,
so not my work.
Yeah, people were from Canada asking this question was from Canada. And then you mentioned that TDP43 was seen independent of stress granules. Is that cleave TDP43 aggregation. Can you please clarify what was specifically seen in those experiments?
So that's a great question. So whether or not it's actually cleaved TDP43 or a modified or a splicing isoform of TDP43, I don't have that data yet. That's coming. We do know that they label with P62, so it's possible that they see terminal fragments, but we don't see any TDP43 aggregation at all in our mouse models. Not in that one anyways.
So there's an interesting question here. So you used increased heat as a way to increase stress, but now the question says, how about people living in hot areas or hot spots in the world? Do you think they may be more susceptible to having those stress granules?
Yeah, so I've received that question many times, and so that's a complicated one. So, I mean, our normal physiology is that we regulate our body temperature, right? And so, and this is an extreme experimental condition where the mice don't have a moment to do that, really. So there is no data that says that there's a higher incidence of TDP43 around the equator or at higher temperature places. There's no data for that. Is that because there's no epidemiology that's being done that's looking at that, or it's really not true? I mean, I can't say. So I want to be very careful that we're not using heat to imply that living in a hot area is linked to ALS, because we really don't have that. We're using it as an experimental stress that's non-invasive, and basically it's what the vets would allow us to do to perturb the mice.
Okay, thank you.
Christine, can I ask you a question? So you've beautifully shown that loss of TDP43 function impairs or leads to the reduction of G3BP1 through two independent mechanisms functionally. Do we know whether loss of G3BP1 in adult motor neurons is sufficient to drive motor neuron defects, cause ALS-like symptoms?
So great question. It's a great question. So I don't have the Crelox, I don't have a floxed allele of G3BP1 to answer that question. We do have the G3BP1 knockout mice are currently breeding, and we'll be actually taking the heterozygotes and mating them into these mice and to see whether or not we drive, we maybe can do that with the G93A and see if we can drive motor neuron loss, accelerate it. The other thing that we're using is the NSP3 peptide, which is a virally encoded peptide that actually sits in the G3BP1 binding pocket, the same as where this cryptic is, and will prevent stress granule formation. This is how Peter Todd did this in the flies. We're going to do it in the mice. It's ongoing right now.
Great.
Hello, nice to talk about these stress granules. So my question is that were these stress granules in a solid state form, or it was just liquid phase that you studied?
So this is mouse tissues. We can't answer that, right? This is fixed tissue from mice, so we can't address the dynamicism with a FRAP experiment. That's not possible.
So, and another question, that these TDP aggregations are occurs inside the stress granules or before making the stress granules?
So where we see the TDP43 cytoplasmic puncta, this is in a model that is not making stress granules.
Thank you.
You're welcome.
Okay, there are no other questions. Let's thank Christine one more time.